Now we're going to discuss lipid metabolism, but we're going to start with catabolism. Remember, metabolism is both catabolism and anabolism. So we're looking at the degradation of fatty acids first. I shared a link with you that shows the hummingbird flying in slow motion. I hope you took a look at that. I'd like to talk about hummingbirds. I'm actually a bird lover, and I feed hummingbirds among other birds in my backyard, but I think these are amazing little creatures. We have them in Minnesota, and they spend their winters in South America. They actually fly across the Gulf of Mexico, and typically that trip is about 457 miles and takes them about 22 hours to complete. So before they begin their migration, they bulk up on body fat. So they increase their body fat by about 70%. Typically, a hummingbird weighs about 1/10 of an ounce. Not very much at all. It's a tiny little thing. At rest, they respire 200 breaths per minute. That's amazing. That's a lot of breathing. High metabolism. Their heart beats at rest 1,260 beats per minute. The metabolism in these little birds is 100 times greater than an elephant's. Their wings beat at 70 beats per second. Goes up to 200 beats per second if they're diving. And they can average 25 to 30 miles per hour. That's incredible. And they cannot use glucose entering the glycolytic pathway to obtain the energy they need for their migration. So they bulk up, and those carbohydrates are converted to lipids or. So lipid metabolism is divided into two sections, catabolism, anabolism. We're going to look at catabolism first in this video. Our primary sources of fatty acids for oxidation are going to be from our diets or mobilization from our cellular stores. Fatty acids are stored as triacylglycerols in adipose tissue. Some of us have more of that than others. I'm one of those. But at any rate, those fatty acids stored as triacylglycerols can be mobilized for use by peripheral tissues. In order to do that, we need a lipase. So lipases are responsible for liberating the individual fatty acids. So these hydrolyze those from the glycerol molecule. Those fatty acids can undergo beta oxidation, and then the glycerol produced by that can be converted to DHAP, which again is a glycolysis metabolite, and can enter metabolism, or enter glycolysis. So how are these fats mobilized? How are they broken down? And are there other ways to oxidize fatty acids? These are some of the questions you should be able to answer after you have covered this material, or we have. So this is a micrograph of an adipose cell or fat cell, and it is filled with globules of triacylglycerols, and they occupy most of the volume of these cells. These triacylglycerols are our principal stored energy reserve. I'm sorry, I am so tongue-tied today. And they're stored in these adipocytes. For an average person of about 154 pounds, the stored metabolic fuel breaks down like this. You don't need to know these numbers, by the way. But fat, adipose tissue, available energy, 555,000 kilojoules. From protein, which we don't really want to break down, we still can generate a lot of energy, 102,000 kilojoules. Glycogen, 1,900 in the muscle, glycogen in the liver, about 1,100, and glucose in extracellular fluids, about 320. So our major stores of energy or available energy are actually in fat and a little less in protein. The calories in our diet from fats are 30 to 60%, depending upon, your intake of fat. But fat accounts for about 80% of our available energy. More fat is stored than carbohydrates. And we saw that. We store about a day's worth of energy in the form of glycogen. So this is a high energy yield per gram of fat compared to carbohydrates. So this is a flow chart, which I like, that shows the regulation or the activation of fatty acid degradation, which is what we're looking at. So again, we've got hormonal regulation. If we have low energy, glucagon and or fight or flight response and secrete epinephrine, each of these hormones is capable of binding a receptor in the cell plasma membrane and activating adenylyl cyclase. This causes the cyclization of ATP to form cyclic AMP. That is our second messenger molecule, and you've seen that before. We just saw that in the regulation of glycogen synthesis or glycogenolysis. So similar method of regulation here. So that cyclic AMP is going to activate a protein kinase, which in turn will phosphorylate this triacylglycerol lipase. It's inactive in its unphosphorylated state, active when it's phosphorylated. The correct name for this enzyme is hormone-sensitive lipase. So once this is activated, we are going to begin hydrolyzing those fatty acids from triacylglycerols. So it will hydrolyze either C1 or C3 of triacylglycerols. There are then subsequent hydrolysis reactions. So we remove one with the hormone-sensitive lipase, and we get a diacylglycerol, or a DAG. DAG lipase will then hydrolyze a second fatty acid, so we end up with a monoacylglycerol. And finally, monoacylglycerol, or MAG lipase, will release that last fatty acid, and then we also have finally our glycerol molecule. So lipases are produce fatty acids in the cytosol of adipocytes. These fatty acids are then activated as CoA derivatives in the mitochondrial cytosol, and then are transported through the inner mitochondrial membrane as carnitine derivatives. And we're going to go through this process and take a look at how this is done. After these fatty acids are transferred into the matrix, they are returned to the CoA derivatives. At this point, they are unable to undergo beta oxidation. So you know that we have different numbers of carbons in our fatty acid chains. We have some that are short, We can have fatty acids with 4 to 8 carbons, for example, and these are short chain fatty acids. We can have up to 12 carbons in a chain. These are medium chain fatty acids. And then our longer chain fatty acids can be up to 16 carbons. So mitochondria exclusively metabolize the 4 to 12 carbon chain fatty acids, where the long chain fatty acids are metabolized in peroxisomes, and they actually prefer 4 carbons in the chain. So before we can transfer that fatty acid, we have to derivatize that with a carnitine. So here we have L-carnitine, and once that fatty acid has been modified with CoA, that does target it for degradation, but in order for it to enter into the mitochondrial matrix, it has to be modified with the carnitine molecule. So the carnitine substitutes for the CoA. This is then transferred, or this transferase, into the mitochondrial matrix as this acyl carnitine. Once it appears in the matrix, then the acyl carnitine, we will transfer that the carbon chain back to the CoA molecule. The carnitine then is reutilized, it's transferred back out of the mitochondrial matrix into that cytosol, or the intermembrane space, and then we can proceed with beta oxidation. So there are 4 reactions, basically, in this cycle. Each round of beta oxidation involves 4 steps. We have an oxidation, a hydration, an oxidation, and a cleavage. And this cycles over and over until that fatty acid has been completely degraded. So the first step, or the first oxidation, we have this fatty acyl CoA. Okay, so here's our fatty acid. We are going to remove hydrogen atoms from the alpha and the beta carbon, so alpha and beta carbon, and form a double bond. At the same time, we are going to reduce FAD to FADH2. The enzyme required here is acyl-CoA dehydrogenase. And what we produce is trans-enol-CoA. Second step is a hydration. We're going to add water across that double bond. Okay? So here's our trans-enol-CoA. We have our double bond between C-alpha and C-beta. We're going to add water across that bond, and we'll get this hydroxyl group on C, or the beta carbon. So we have now produced beta hydroxyl acyl CoA, and the enzyme here is enol CoA hydratase. So this is our hydration reaction. Now we're going to undergo a second oxidation reaction. We are going to oxidize that beta hydroxyl acyl CoA to a beta keto acyl CoA. And at the same time, we're going to reduce NAD plus, or NAD plus, to NADH plus a hydrogen ion. The enzyme involved here is 3-hydroxyl-AC-CoA dehydrogenase. And the fourth reaction in this cycle is a cleavage reaction. we are going to cleave an acyl unit and produce acetyl-CoA, which then can be utilized in the citric acid cycle. So what we have is our beta-ketoacyl-CoA. We have CoASH, so we need another one of those. And we're going to transfer this group to our new CoA molecule and release our acetyl-CoA. So we shorten our fatty acyl-CoA by 1 acyl unit, and thiolase is the enzyme that is required here. We are making a thioester linkage. So let's look at the beta oxidation of a 14-carbon amino acid, myristic acid, or not amino acid, sorry, fatty acid. So this 14-carbon lipid will undergo these 4 reactions until we have completely broken down that lipid chain. What we get from that, ultimately, I should say, after 6 cycles, are 7 acetyl-CoA molecules. At the same time, we will produce NADH2, or NADH, FADH2, and acetyl-CoA. So for every the number of beta oxidation cycles depends upon the length of fatty acid. It is always 1 less than the number of acyl-CoA groups formed. So if we have, a 12-carbon fatty acid, we are going to divide that by 2, and that tells us how many CoAs we are going to get, or acetyl-CoAs we're going to get. And the oxidation cycles is always going to be 1 less than the number of acetyl-CoAs that we get. So a 12-carbon fatty acid will undergo 5 cycles. An 18-carbon fatty acid will undergo 8 cycles. And lastly, we're looking at the beta oxidation of 1 palmitic acid fatty acid. So this is just to emphasize the number of ATP molecules that are generated with beta oxidation. So if you remember, palmitic acid is a 16-carbon lipid or fatty acid. So we will get 8 acetyl-CoAs from that. Here we go. So we have our 8 acetyl-CoAs. But we get FADH2, 7 NADH, plus 7 hydrogen ions. These can all go into the electron transport system or chain and contribute to oxidative phosphorylation and the synthesis of ATP, from which we get 100 ATP. Let's see where else we're going here. In the citric acid cycle, we get 8 ATP. So, we've got to remember we're going to use 2 ATP somewhere because in total we get 106 ATP molecules. Okay. So there's a very large energy yield with respect to the reduced, and it comes from the highly reduced state of the carbon in fatty acids. So this makes fatty acids the fuel of choice for birds that undergo migration and animals that also hibernate. And we'll look at, well, actually, we already looked at polar bears. So very important energy molecule as well. This concludes our discussion of catabolism. Now we're going to move on to fatty acid synthesis. Oh, sorry, there's one more. Excuse me. So this is just a table that accentuates the free energy, I'm sorry, that is obtained, as well as the yield of ATP. So free energy from beta oxidation, in total, 3,233 kilojoules per mole and 106 ATP molecules. Pretty incredible.